Welcome to Flourish. I'm Diane Planidin, and you're in the right place if you're ready to create inspired life. And we do so by working on our own personal development so we can be strong role models for those we love and mentor and keep a strong mindset for ourselves. Today, we are on week 3, chapter 15 in our journey through Psych-100 at Queen's University. So let's get started. Chapter 15 is about hormones and behavior. The goal of this module is to introduce you to the topic of hormones and behavior. This field of study is also called behavioral endocrinology, which is a scientific study of the interaction between hormones and behavior. This interaction is bidirectional. Hormones can influence behavior, and behavior can sometimes influence hormone concentrations. Hormones are chemical messengers released from the endocrine glands that travel through the blood system to influence the nervous system to regulate behaviors such as aggression, mating, and parenting of individuals. Learning objectives. Define the basic terminology and basic principles of hormone behavior interactions. Explain the role of hormones in behavioral sex differentiation. Explain the role of hormones in aggressive behavior. Explain the role of hormones in parental behavior, and provide examples of some common hormone behavior interactions. As mentioned previously, I am not a teacher, I am a student, and I'm sharing my learning journey with you as I research what we're all about for my book. Here we go. Introduction. This module describes the relationship between hormones and behavior. Many readers are likely already familiar with the general idea that hormones can affect behavior. Students are generally familiar with the idea that sex hormone concentrations increase in the blood during puberty and decrease as we age, especially after about 50 years of age. Sexual behavior shows a similar pattern. Most people also know about the relationship between aggression and anabolic steroid hormones, and they know that administration of artificial steroid hormones sometimes results in uncontrollable, violent behavior called roid rage. Many different hormones can influence several types of behavior, but for the purposes of this module, we will restrict our discussion to just a few examples of hormones and behaviors. For example, are behavioral sex differences the result of hormones, the environment, or some combination of factors? Why are men much more likely than women to commit aggressive acts? Are hormones involved in mediating the so-called maternal instinct? Behavioral endocrinologists are interested in how the general physiological effects of hormones alter the development and expression of behavior, and how behavior may influence the effects of hormones. This module describes both phenomenologically and functionally how hormones affect behavior. To understand the hormone behavior relationship, it is important briefly to describe hormones. Hormones are organic chemical messengers produced and released by specialized glands called endocrine glands. Hormones are released from these glands into the blood, where they may travel to act on target structures at some distance from their origin. Hormones are similar in function to neurotransmitters, the chemicals used by the nervous system in coordinating animals' activities. However, hormones can operate over a greater distance and over a much greater temporal range than neurotransmitters. Examples of hormones that influence behavior include steroid hormones, such as testosterone, estradiol, progesterone, and cortisol. Several types of protein or peptide hormones also influence behavior, including oxytocin, vasopressin, prolactin, and leptin. Focus topic number 1, neural transmission versus hormonal communication. Although neural and hormonal communication both rely on chemical signals, several prominent differences exist. Communication in the nervous system is analogous to traveling on a train. You can use the train in your travel plans as long as tracks exist between your proposed origin and destination. Likewise, neural messages can travel only to destinations along existing nerve tracks. Hormonal communication, on the other hand, is like traveling in a car. You can drive to many more destinations than train travel allows because there are many more roads than railroad tracks. Similarly, hormonal messages can travel anywhere in the body via the circulatory system. Any cell receiving blood is potentially able to receive a hormonal message. Neural and hormonal communication differ in other ways as well. To illustrate them, consider the difference between digital and analog technology. Neural messages are digital, all in one events that have rapid onset and offset. Neural signals can take place in milliseconds. Accordingly, the nervous system mediates changes in the body that are relatively rapid. For example, the nervous system regulates immediate food intake and directs body movement. In contrast, hormonal messages are analog, graded events that may take seconds, minutes, or even hours to occur. Hormones can mediate long-term process such as growth, development, reproduction, and metabolism. Hormonal and neural messages are both chemical in nature and they are released and received by cells in a similar manner. However, there are important differences as well. Neurotransmitters, the chemical messengers used by neurons, travel a distance of only 20 to 30 nanometers to the membrane of the postsynaptic neuron, where they bind with receptors. Hormones enter the circulatory system and may travel from 1 millimeter to 2 meters before arriving at a target cell, where they bond with specific receptors. Another distinction between neural and hormonal communication is the degree of voluntary control that can be exerted over their functioning. In general, there is more voluntary control of neural than hormonal signals. It is virtually impossible to will a change in your thyroid hormone levels, for example, whereas moving your limbs on command is easy. Although these are significant differences, the division between the nervous system and the endocrine system is becoming more blurred as we learn more about how the nervous system regulates hormonal communication. A better understanding of the interface between the endocrine system and the nervous system, called neuroendocrinology, is likely to yield important advances in the future study of the interaction between hormones and behavior. We have a table here of prominent hormones that influence behavior. Steroid hormones, cortisol, increases carbohydrate metabolism, mediates stress responses. Estradiol, uterine and other female tissue development, regulates sexual motivation and performance in females and males. Testosterone promotes sperm production and male secondary sexual characteristics, promotes sexual motivation and behavior, typically by being converted to estradiol. Peptides and protein hormones. Oxytocin stimulates milk letdown and uterine contractions during birth, promotes social bonding. Prolactin, many actions relating to reproduction, water balance, and behavior associated with parental care. Thyroxine increases oxidation rates in tissue and affects neural development. Vasopressin increases water reabsorption in the kidney and affects learning and memory. Hormones coordinate the physiology and behavior of individuals by regulating, integrating, and controlling bodily functions. Over evolutionary times, hormones have often been co-opted by the nervous system to influence behavior to ensure reproductive success. For example, the same hormones, testosterone and estradiol, that cause gamete maturation also promote mating behavior. This dual hormonal function ensures that mating behavior occurs when animals have mature gametes available for fertilization. Another example of endocrine regulation of physiological and behavioral function is provided by pregnancy. Estrogens and progesterone concentrations are elevated during pregnancy, and these hormones are often involved in mediating maternal behavior in the mothers. Not all cells are influenced by each and every hormone. Rather, any given hormone can directly influence only cells that have specific hormone receptors for that particular hormone. Cells that have these specific receptors are called target cells for the hormone. The interaction of a hormone with its receptor begins a series of cellular events that eventually lead to activation of enzymatic pathways, or alternatively, turns on or turns off gene activation that regulates protein synthesis. The newly synthesized proteins may activate or deactivate other genes, causing yet another cascade of cellular events. Importantly, sufficient number of appropriate hormone receptors must be available for a specific hormone to produce any effects. For example, testosterone is important for male sexual behavior. If men have too little testosterone, then sexual motivation may be low and it can be restored by testosterone treatment. However, if men have normal or even elevated levels of testosterone, yet display low sexual drive, then it might be possible for a lack of receptors to be the cause, and treatment with additional hormones will not be effective. How might hormones affect behavior? In terms of their behavior, one can think of humans and other animals conceptually as comprised of three interacting components. 1. Input systems, 2. Integrators, and 3. Output systems. Hormones do not cause behavioral changes. Rather, hormones influence these three systems so that the specific stimuli are more likely to elicit certain responses in the appropriate behavioral or social context. In other words, hormones change the probability that a particular behavior will be emitted in the appropriate situation. This is a critical distinction that can now affect how we think of hormone behavior relationships. We can apply this three-component behavioral scheme to a simple behavior, singing in zebra finches. Only male zebra finches sing. If the testes of adult male finches are removed, then the birds reduce singing, but castrated finches resume singing if the testes are reimplanted, or if the birds are treated with either testosterone or estradiol. Although we commonly consider androgens to be male hormones and estrogens to be female hormones, it is common for testosterone to be converted to estradiol in nerve cells. Thus, many male-like behaviors are associated with the actions of estrogens. Indeed, all estrogens must first be converted from androgens because of the typical biochemical synthesis process. If the converting enzyme is low or missing, then it is possible for females to produce excessive androgens and subsequently develop associated male traits. It is also possible for estrogens in the environment to affect the nervous system of animals, including people. Again, singing behavior is most frequent when blood testosterone or estrogen concentrations are high. Males sing to attract mates or ward off potential competition from their territories. Although it is apparent from these observations that estrogens are somehow involved in singing, how might the three-component framework just introduced help us formulate hypotheses to explore estrogen's role in this behavior? By examining input systems, we could determine whether estrogens alter the bird's sensory capabilities, making the environmental cues that normally elicit singing more salient. If this were the case, then females or competitors might be more easily seen or heard. Estrogens also could influence the central nervous system. Neuronal architecture or the speed of neural processing could change in the presence of estrogens. Higher neural processes also might be influenced. Finally, the effector organs, muscles in this case, could be affected by the presence of estrogens. Blood estrogen concentrations might somehow affect the muscles of a songbird's syrinx, the vocal organ of birds. Estrogens, therefore, could affect a bird song by influencing the sensory capabilities, central processing system, or effector organs of an individual bird. We do not understand completely how estrogen, derived from testosterone, influences birdsong, but in most cases, hormones can be considered to affect behavior by influencing one, two, or all three of these components. And this three-part framework can aid in the design of hypotheses and experiments to explore these issues. How might behaviors affect hormones? The bird song example demonstrates how hormones can affect behavior, but as noted, the reciprocal relation also occurs. That is, behavior can affect hormone concentrations. For example, the sight of a territorial intruder may elevate blood testosterone concentrations in resident male birds and thereby stimulate singing or fighting behavior. Similarly, male mice or rhesus monkeys that lose a fight decrease circulating testosterone concentrations for several days or even weeks afterwards. Comparable results have also been reported in humans. Testosterone concentrations are affected not only in humans involved in physical combat, but also in those involved in simulated battles. For example, testosterone concentrations were elevated in winners and reduced in losers of regional chess tournaments. People do not have to be directly involved in a contest to have their hormones affected by the outcome of the contest. Male fans of both the Brazilian and Italian teams were recruited to provide saliva samples to be assayed for testosterone before and after the final game of the World Cup soccer match in 1994. Brazil and Italy were tied going into the final game, but Brazil won on a penalty kick at the last possible moment. The Brazilian fans were elated, and the Italian fans were crestfallen. When the samples were assayed, 11 of 12 Brazilian fans who were sampled had increased testosterone concentrations, and 9 of 9 Italian fans had decreased testosterone concentrations compared with pre-game baseline values. In some cases, hormones can be affected by anticipation of behavior. For example, testosterone concentrations also influence sexual motivation and behavior in women. In one study, the interaction between sexual intercourse and testosterone was compared with other activities in women. On 3 separate occasions, women provided a pre-activity, post-activity, and next morning saliva sample. After analysis, the women's testosterone was determined to be elevated prior to intercourse as compared to other times. Thus, an anticipatory relationship exists between sexual behavior and testosterone. Testosterone values were higher post intercourse compared to exercise, suggesting that engaging in sexual behavior may also influence hormone concentrations in women. Sex differences. Hens and roosters are different. Cows and bulls are different. Men and women are different. Even girls and boys are different. Humans, like many animals, are sexually dimorphic in the size and shape of their bodies, their physiology, and for our purposes, their behavior. Behavior of boys and girls differs in many ways. Girls generally excel in verbal abilities relative to boys. Boys are nearly twice as likely as girls to suffer from dyslexia and stuttering, and nearly 4 times more likely to suffer from autism. Boys are generally better than girls at tasks that require visuospatial abilities. Girls engage in nurturing behaviors more frequently than boys. More than 90% of all anorexia nervosa cases involve young women. Young men are twice as likely as young women to suffer from schizophrenia. Boys are much more aggressive and generally engage in more rough and tumble play than girls. Many sex differences, such as the difference in aggressiveness, persist throughout adulthood. For example, there are many more men than women serving prison sentences for violent behavior. The hormonal differences between men and women may account for adult sex differences that develop during puberty. But what accounts for behavioral sex differences among children prior to puberty and activation of their gonads? Hormonal secretion from the developing gonads determine whether the individual develops in a male or female manner. The mammalian embryonic testes produce androgens, as well as peptide hormones that steer the development of the body, central nervous system, and subsequent behavior in a male direction. The embryonic ovaries of mammals are virtually quiescent and do not secrete high concentrations of hormones. In the presence of ovaries, or in the complete absence of any gonads, morphological, neural, and later behavioral development follows a female pathway. Gonadal steroid hormones have organizational effects upon brain and behavior. The organizing effects of steroid hormones are relatively constrained to the early stages of development. An asymmetry exists in the effects of testes and ovaries on the organization of the behavior in mammals. Hormone exposure early in life has organizational effects on subsequent rodent behavior. Early steroid hormone treatment causes relatively irreversible and permanent masculinization of rodent behavior. These early hormone effects can be contrasted with the reversible behavioral influence of steroid hormones provided in adulthood, which are called activational effects. The activational effects of hormones on adult behavior are temporary and may wane soon after the hormone is metabolized. Thus, typical male behavior requires exposure to androgens during gestation in humans or immediately after birth in rodents to somewhat masculinize the brain and also requires androgens during or after puberty to activate these neural circuits. Typical female behavior requires a lack of exposure to androgens early in life, which leads to feminization of the brain and also requires estrogens to activate these neural circuits in adulthood. But this simple dichotomy, which works well with animals with very distinct sexual dimorphisms in behavior, has many caveats when applied to people. If you walk through any major toy store, then you will likely observe a couple of aisles filled with pink boxes and the complete absence of pink packaging of toys in adjacent aisles. Remarkably, you will also see a strong self-segregation of boys and girls in these aisles. It is rare to see boys in the pink aisles and vice versa. The toy manufacturers are often accused of making toys that are gender biased, but it seems more likely that boys and girls enjoy playing with specific types and colors of toys. Indeed, toy manufacturers would immediately double their sales if they could sell toys to both sexes. Boys generally prefer toys such as trucks and balls, and girls generally prefer toys such as dolls, although it is doubtful that there are genes that encode preferences for toy cars and trucks on the Y chromosome. It is possible that hormones might shape the development of a child's brain to prefer certain types of toys or styles of play behavior. It is reasonable to believe that children learn which type of toys and which style of play are appropriate to their gender. How can we understand and separate the contribution of physiological mechanisms from learning to understand sex differences in human behavior? To untangle these issues, animal models are often used. Unlike the situation in humans, where sex differences are usually only a matter of degree, often slight, in some animals, members of only one sex may display a particular behavior. As noted, often only male songbirds sing. Studies of such strong sex-biased behaviors are particularly valuable for understanding the interaction among behavior, hormones, and the nervous system. A study of vervet monkeys calls into question the primacy of learning in the establishment of toy preferences. Female vervet monkeys preferred girl-typical toys such as dolls or cooking pots, whereas male vervet monkeys preferred boy-typical toys such as cars or balls. There were no sex differences in preference for gender-neutral toys, such as picture books or stuffed animals. Presumably, monkeys have no prior concept of boy or girl toys. Young rhesus monkeys are also shown similar toy preferences. What then underlies the sex difference in toy preference? It is possible that certain attributes of toys or objects appeal to either boys or girls. Toys that appeal to boys or male vervet or rhesus monkeys, in this case, a ball or toy car, are objects that can be moved actively through space, toys that can be incorporated into active, rough, and tumble play. The appeal of toys that girls or female vervet monkeys prefer appears to be based on color. Pink and red may provoke attention to infants. Society may reinforce such stereotypical responses to gender-typical toys. The sex differences in toy preferences emerge by 12 or 24 months of age and seem fixed by 36 months of age. But are sex differences in toys preferences present during the first year of life? It is difficult to ask pre-verbal infants what they prefer. But in studies where investigators examined the amount of time that babies looked at different toys, eye tracking data indicate that infants as young as 3 months showed sex differences in toy preferences. Girls preferred dolls, whereas boys preferred trucks. Another result that suggests, but does not prove, that hormones are involved in toy preferences is the observation that girls diagnosed with congenital adrenal hyperplasia, whose adrenal glands produce varying amounts of androgens early in life, played with masculine toys more often than girls without CAH. Further, a dose-response relationship between the extent of the disorder, i.e., degree or fetal androgen exposure, and degree of masculinization of play behavior was observed. Are the sex differences in toy preferences or play activity, for example, the inevitable consequences of the differential endocrine environments of boys and girls, or are these differences imposed by cultural practices and beliefs? Are these differences the result of receiving gender-specific toys from an early age, or are these differences some combination of endocrine and cultural factors? Again, these are difficult questions to unravel in people. Even when behavioral sex differences appear early in development, there seems to be some question regarding the influences of societal expectations. One example is the pattern of human play behavior during which males are more physical. This pattern is seen in a number of other species, including non-human primates, rats, and dogs. Is the difference in the frequency of rough and tumble play between boys and girls due to biological factors associated with being male or female, or is it due to cultural expectations and learning? If there's a combination of biological and cultural influences mediating the frequency of rough and tumble play, then what proportion of variation between the sexes is due to biological factors and what proportion is due to social influences? Importantly, is it appropriate to talk about normal sex differences when these traits virtually always arrange themselves along a continuum rather than in discrete categories? Sex differences are common in humans and in non-human animals. Because males and females differ in the ratio of androgenic and estrogenic steroid hormone concentrations, behavioral endocrinologists have been particularly interested in the extent to which behavioral sex differences are mediated by hormones. The process of becoming female or male is called sexual differentiation. The primary step in sexual differentiation occurs at fertilization. In mammals, the ovum can be fertilized by sperm bearing either a Y or an X chromosome. This process is called sex determination. The chromosomal sex of homogenetic mammals, XX, is female. The chromosomal sex of heterogametic mammals, XY, is male. Chromosomal sex determines gonadal sex. Virtually all subsequent sexual differentiation is typically the result of differential exposure to gonadal steroid hormones. Thus, gonadal sex determines hormonal sex, which regulates morphological sex. Morphological differences in the central nervous system, as well as in some effector organs such as muscles, leads to behavioral sex differences. The process of sexual differentiation is complicated and the potential for errors is present. Perinatal exposure to androgens is the most common cause of anomalous sexual differentiation among females. The source of androgen may be internal or external. Turner syndrome results when the second X chromosome is missing or damaged. These individuals possess dysgenic ovaries and not exposed to steroid hormones until puberty. Interestingly, women with Turner syndrome often have impaired spatial memory. Female mammals are considered the neutral sex. Additional physiological steps are required for male differentiation, and more steps bring more possibilities for errors in differentiation. Some examples of male anomalous sexual differentiation include 5A reductase deficiency and androgen insensitivity syndrome, or TFM. By studying individuals who do not neatly fall into the dichotic boxes of female or male and for whom the process of sexual differentiation is atypical, behavioral endocrinologists glean hints about the process of typical sexual differentiation. We may ultimately want to know how hormones mediate sex differences in the human brain and behavior. To understand the mechanisms underlying sex differences in the brain and behavior, we return to the bird song example. Birds provide the best evidence that behavioral sex differences are the result of hormonally induced structural changes in the brain. In contrast to mammals, in which structural differences in neural tissues have not been directly linked to behavior, structural differences in avian brains have been directly linked to a sexually behavior. Birdsong. Several brain regions in songbirds display significant sex differences in size. Two major brain circuit pathways, the song production motor pathway and the auditory transmission pathway, have been implicated in learning and production of birdsong. Some parts of the song production pathway of male zebra finches are 3 to 6 times larger than those of female conspecifics. The larger size of these brain areas reflects that neurons in these nuclei are larger, more numerous, and farther apart. Although castration of adult male birds reduces singing, it does not reduce the size of the brain nuclei controlling song production. Similarly, androgen treatment of adult female zebra finches does not induce changes either in singing or in the size of the song control regions. Thus, activational effects of steroid hormones do not account for the sex differences in singing behavior or brain nucleus size in zebra finches. The sex differences in these structures are organized or programmed in the egg by estradiol, or the lack of steroids. Taken together, estrogens appear to be necessary to activate the neural machinery underlying the song system in birds. The testes of birds primarily produce androgens which enter the circulation. The androgens enter neurons containing aromatase, which converts them to estrogens. Indeed, the brain is the primary source of estrogens, which activate masculine behaviors in many bird species. Sex differences in human brain size have been reported for years. More recently, sex differences in specific brain structures have been discovered. Sex differences in a number of cognitive functions have also been reported. Females are generally more sensitive to auditory information, whereas males are more sensitive to visual information. Females are also typically more sensitive than males to taste and olfactory input. Women display less lateralization of cognitive functions than men. On average, females generally excel in verbal, perceptual, and fine motor skills, whereas males outperform females on quantitative and visuospatial tasks, including map reading and direction finding. Although reliable sex differences can be documented, these differences in ability are slight. It is important to note that there is more variation within each sex than between the sexes for most cognitive abilities. Aggressive behaviors. The possibility for aggressive behavior exists whenever the interests of two or more individuals are in conflict. Conflicts are most likely to arise over limited resources such as territories, food, and mates. A social interaction decides which animal gains access to the contested resources. In many cases, a submissive posture or gesture on the part of one animal avoids the necessity of actual combat over a resource. Animals may also participate in threat displays or ritualized combat in which dominance is determined but no physical damage is inflicted. There's an overwhelming circumstantial evidence that androgenic steroid hormones mediate aggressive behaviors across many species. First, seasonal variations in blood plasma concentrations of testosterone and seasonal variation in aggression coincide. For instance, the incidence of aggressive behavior peaks for male deer in autumn when they are secreting high levels of testosterone. Second, aggressive behaviors increase at the time of puberty when the testes become active and blood concentrations of androgens rise. Juvenile deer do not participate in fighting during the mating season. Third, in any given species, males are generally more aggressive than females. This is certainly true of deer. Relative to stags, female deer rarely display aggressive behavior, and their rare aggressive acts are qualitatively different from the aggressive behavior of aggressive males. Finally, castration typically reduces aggression in males, and testosterone replacement therapy restores aggression to pre-castration levels. There are some interesting exceptions to these general observations that are outside the scope of this module. As mentioned, males are generally more aggressive than females. Certainly, human males are much more aggressive than females. Many more men than women are convicted of violent crimes in North America. The sex differences in human aggressiveness appear very early. At every stage throughout the school year, many more boys than girls initiate physical assaults. Almost everyone will acknowledge the existence of this sex difference, but assigning a cause to behavioral sex differences in humans always elicits much debate. It is possible that boys are more aggressive than girls because androgens promote aggressive behavior, and boys have a higher blood concentration of androgens than girls. It is possible that boys and girls differ in their aggressiveness because the brains of boys are exposed to androgens prenatally, and the wiring of their brain is thus organized in a way that facilitates the expression of aggression. It is also possible that boys are encouraged and girls are discouraged by family, peers, or other from acting in an aggressive manner. These three hypotheses are not mutually exclusive, but it is extremely difficult to discriminate among them to account for sex differences in human aggressiveness. What kind of studies would be necessary to assess these hypotheses? It is usually difficult to separate out the influences of environment and physiology on the development of behavior in humans. For example, boys and girls differ in their rough and tumble play at a very young age, which suggests an early physiological influence on aggression. However, parents interact with their male and female offspring differently. They usually play more roughly with male infants than with females, which suggests that the sex difference in aggressiveness is partially learned. This difference in parental interaction style is evident by the first week of life. Because of these complexities in the factor influencing human behavior, the study of hormonal effects on sex differentiated behavior has been pursued in non human animals for which environmental influences can be held relatively constant. Animal models for which sexual differentiation occurs postnatally are often used so that this process can be easily manipulated experimentally. Again, with the appropriate animal model, we can address the questions posed above. Is the sex difference in aggression due to higher adult blood concentration of androgens in males than in females? Or are males more aggressive than females because their brains are organized differently by perinatal hormones? Are males usually more aggressive than females because of an interaction of early and current blood androgen concentration? If male mice are castrated prior to their sixth day of life, then treated with testosterone propionate in adulthood, they show low levels of aggression. Similarly, females overreactomized prior to their sixth day but given androgens in adulthood do not express male-like levels of aggression. Treatment of the perinatally gonadectomized males or females with testosterone prior to their sixth day of life and also in adulthood results in a level of aggression similar to that observed in typical male mice. Thus, in mice, the proclivity for males to act more aggressively than females is organized perinatally by androgens, but also requires the presence of androgens after puberty in order to be fully expressed. In other words, aggression in male mice is both organized and activated by androgens. Testosterone exposure in adulthood without prior organization of the brain by steroid hormones does not evoke typical male levels of aggression. The hormonal control of aggressive behavior in house mice is thus similar to the hormonal mediation of heterosexual male mating behavior in other rodent species. Aggressive behavior is both organized and activated by androgens in many species, including rats, hamsters, voles, dogs, and possibly some primate species. Parental behaviors. Parental behavior can be considered to be any behavior that contributes directly to the survival of fertilized eggs or offspring that have left the body of the female. There are many patterns of mammalian parental care. The developmental status of the newborn is an important factor driving the type and quality of parental care in species. Maternal care is much more common than paternal care. The vast majority of research on hormonal correlates of mammalian parental behavior has been conducted on rats. Rats bear altricial young, and mothers perform a cluster of stereotype maternal behaviors, including nest building, crouching over the pups to allow nursing and to provide warmth, pup retrieval, and increased aggression directed at intruders. If you expose non-pregnant female rats to pups, their most common reaction is to huddle far away from them. Rats avoid new things, neophobia. However, if you expose adult rats to pups every day, they soon begin to behave maternally. This process is called concavation or sensitization, and it appears to serve to reduce the adult rat's fear of pups. Of course, a new mother needs to act maternal as soon as her offspring arrive, not in a week. The onset of maternal behavior in rats is mediated by hormones. Several methods of study, such as hormone removal and replacement therapy, have been used to determine the hormonal correlates of rat maternal behavior. A fast decline of blood concentrations of progesterone in late pregnancy after sustaining high concentrations of this hormone, in combination with high concentrations of estradiol and probably prolactin and oxytocin, induces female rats to behave maternally almost immediately in the presence of pups. This pattern of hormones at parturition overrides the usual fear response of adult rats toward pups, and it permits the onset of maternal behavior. Thus, the so-called maternal instinct requires hormones to increase the approach tendency and lower the avoidance tendency. Laboratory strains of mice and rats are usually docile, but mothers can be quite aggressive towards animals that venture too close to their litter. Progesterone appears to be the primary hormone that induces this maternal aggression in rodents, but species differences exist. The role of maternal aggression in women's behavior has not been adequately described or tested. A series of elegant experiments by Alison Fleming and her collaborators studied the endocrine correlates of the behavior of human mothers as well as the endocrine correlates of maternal attitudes as expressed in self-report questionnaires. Responses such as patting, cuddling, or kissing the baby were called affectionate behaviors. Talking, singing, or cooing to the baby were considered vocal behaviors. Both affectionate and vocal behaviors were considered approach behaviors. Basic caregiving activities such as changing diapers and burping the infants were also recorded. In these studies, no relationship between hormone concentrations and maternal responsiveness, as measured by attitude questionnaires, was found. For example, most women showed an increasing positive self-image during pregnancy that dipped during the second half of pregnancy but recovered after parturition. A related dip in feelings of maternal engagement occurred during late pregnancy but rebounded substantially after birth in most women. However, when behavior rather than questionnaire responses was compared with hormone concentrations, a different story emerged. Blood plasma concentrations of cortisol were positively associated with approach behaviors. In other words, women who had high concentration of blood cortisol in samples obtained immediately before or after nursing engaged in more physically affectionate behaviors and talked more often to their babies than mothers with low cortisol concentrations. Additional analysis from this study revealed that the correlation was even greater for mothers that had reported positive maternal regard during gestation. Indeed, nearly half of the variation in maternal behavior among women could be accounted for by cortisol concentrations and positive maternal attitudes during pregnancy. Presumably, cortisol does not induce maternal behaviors directly, but it may act indirectly on the quality of maternal care by evoking an increase in the mother's general level of arousal, thus increasing her responsiveness to infant-generated cues. New mothers with high cortisol concentrations were also more attracted to their infants' odors, were superior in identifying their infants, and generally found cues from infants highly appealing. The medial or preoptic areas is critical for expression of rat maternal behavior. The amygdala appears to totonically inhibit the expression of maternal behavior. Adult rats are fearful of pups, a response that is apparently mediated by chemosensory information. Lesions of the amygdala are afferent sensory pathways from the vomeronasal organ to the amygdala disinhibit the expression of maternal behavior. Hormones or sensitization likely act to disinhibit the amygdala, thus permitting the occurrence of maternal behavior. Although correlations have been established, direct evidence of brain structural changes in human mothers remains unspecified. Considered together, there are many examples of hormones influencing behavior and of behavior feeding back to influence hormone secretion. More and more examples of hormone behavior interactions are discovered, including hormones in the mediation of food and fluid intake, social interactions, salt balance, learning and memory, stress coping, as well as psychopathology, including depression, anxiety disorders, eating disorders, postpartum depression, and seasonal depression. Additional research should reveal how these hormones, behaviors, interactions are mediated. Well, that was quite the topic, wasn't it? Really, truly quite fascinating. And if you like the show, share it with someone you know and maybe hit that subscribe button. Give me a thumbs up or even some comments. I did struggle again in this chapter on pronunciation, but I did as best as I could. So if you have any helpful hints, yeah, put it in the comments. I'd love to hear from you. You deserve to pass this course, and you deserve to be a success that you already know you can be, and you deserve to live a more inspired life.